All right, welcome back everyone. My name is Kevin Libowitz. I help to co-chair the AI PHA4GE working group. And I hope you guys enjoyed your tea break and poster sessions. In the Priority Pathogen Diseases Part 2, we will be continuing the discussion that was introduced in Part 1. So this session highlights how open data, best practices, and community-driven initiatives are transforming our ability to monitor and respond to infectious diseases across the world. Similar to the last session, we're going to have 5 speakers speak in series, and then we'll be closing and getting all the speakers back up for a 20-minute Q&A session. So we will be beginning with our first speaker, Gültekin Ünal, who will be speaking about his work being done in the PHAGE's Bioinformatics Pipeline and Visualizations Working Group. So please welcome Gültekin to the stage. Thank you all.
So, hello everybody. This is Gültekin from Türkiye. Let me find my... okay. Hello again. This is Gültekin from Türkiye. I was working for Ankara University Faculty of Veterinary Medicine, Department of Microbiology as a postdoc, but I recently started to work for WHO Country Office for Türkiye as a National Professional Officer for laboratories. So today I will talk about some of the works that has been done by us in the PHAGE, Bioinformatics Pipelines and Data Visualization Working Group. So I know it's a bit late. It's 4 p.m. and I don't want to burden you with like a lot of information, okay? So I would like to start with empathy. So I want you to ruminate this word throughout my talk, okay? So why I'm talking about empathy? Because let me give you my example about this. So I was interested in bioinformatics all the time, and there were not many people around me to help me on this idea. And I always look for others to share and discuss the things about the bioinformatics with me. I was struggling a lot because in Turkey we can't find a lot of people dealing with, especially with the microbial genomic stuff. So I was thinking that there might be some people around the world that I can discuss and find solutions about the problems that I'm having. So I looked internet, I checked everything, and I found PHAGE. It was around 2020, I think. And I saw that we need to know always there's someone else having almost the same problems as you're having and the same challenges towards. So, as I said, empathy helped me to find PHAGE and enter PHAGE as a member. So, which connected me with the people that feeling the same with me, feeling the same way as me. So in the working group, on the other hand, we are focusing mostly on the lived experiences and real needs. We are not trying to discover the USA again. This is a word that we are using in Turkey all the time. We don't need to rediscover the USA. And this is creating a starting point for developing collaborative solutions that directly supports the public health. So what is the problem that we are trying to solve here? The problem is bioinformatics is powerful. We know that, we saw that, we are feeling it, but it's still fragmented. We had a lot of talks prior to my presentation. There are many pipelines out there. There are independent, some of them institutionalized, but there's an incompatibility problem in that, and some of them using different file formats. So reproducibility, scalability, and portability remain some of the major challenges we have. And we talked about the metadata, of course. We talked about the workforce, resource inequity, which creates barriers to adopt these strategies. Governance gaps, another topic, and the version control reduce sustainability of our resources. So what PHA4GE offers had been explained by Professor Allen. I'm not going to mention about that, but I will talk about what we offer to the community. So we are mostly focusing on the concepts that I mentioned, like interoperability, reproducibility, sustainability across different pipelines, right? So this includes the QC, metadata practice, versioning, open source, licensing, and strong documentation is one of the biggest impact. So the goal is simple. We are, of course, in an ideal world, trying to enable every laboratory, every institution to run reliable genomic analysis, and regardless of resources they have. And we're trying to do it with open, interoperable, and sustainable way. So how many people we have in our working group? We have more than 150 members, and we are thinking that we are one of the biggest working group at PHA4GE. I'm not sure about that. We can calculate that again. And great numbers come with great responsibilities, we know that. So how we do it? I would like to start with the brainstorm. We are having monthly meetings now in our working group. So with the members, we are gathering together and talking about the stuff that we are facing, some of the problems, and we are sharing ideas together, some inputs from the participants we have. Then we are collecting these ideas, and if anything comes up for us to go ahead, we are taking that idea and going to the next stage, which is research and data dump. So basically, we are going into the Google Drive, opening a folder, and putting everything in it, every evidence, every data sets, every paper, every information there. Then we are then shaping it to create a conceptual, finalized draft. So I'm talking about these things very fastly, but this is not happening at that fast. Then finally, after finalizing the draft, we are going into the step that we are gathering some feedback from the community, which is we are trying to get in touch with the domain experts, some people that might be users of the solutions that we are trying to provide here. After we have done all of these, we are going into the field that we sharing this document, which is sometimes, I mean, most of the times happening as a guidance document. We share it in our GitHub page. So if anybody interested in, you can scan the QR code and go to the resources that we have so far. Also, we have the link. You can scan it, or otherwise you can find me somewhere during the conference, and I can guide you through that. So some of the things that had been done by the previous co-chairs of our working group, who is Kevin Lewitt and Jamie Southgate. They have done an amazing job, and they created some of the guidance documents for our working group, which are bioinformatics solutions for SARS-CoV-2 genomic analysis, QC solutions for SARS-CoV-2, identifying SARS-CoV-2 recombinants, and bioinformatics solutions for mpox genomics analysis. But what we have done after taking over in 2023, we started with different sub-working groups. So we have working group, the main one. Then we have sub-working groups are working on specific subjects, like wastewater. So wastewater is one of them. So Emily Smith and Josh Levy put a lot of effort into this and created wastewater surveillance guidance documents. It has strategies, data analysis, data sharing ethics and legalities, alongside with case studies. It's on the GitHub page, and you can take a look at that. And we have HIV bioinformatics solutions. These are the things that included in our guidance documents, from introduction to the case studies by Amy, Mark, Frank, and Abbas. We also have another one. These are the guidance documents that I'm talking about, by the way. That's another one. This is the 10 best practices for public health. So the leading by Kevin again, by many others. I can't read all the names, but if you Google it, you will find it. The idea is to create some 10 best standardized approach to create bioinformatics pipelines and tools. So they came up with 10 simple ones, and this is from the publicly accessible repositories, being open science, open source license, common file formats, and testing, and reference data requirements, containerization, and workflow management systems are some of them. What else we had? We had influenza guidance documents with the Emily again leading on that. We worked on the influenza guidance documents, starting from the genomic structure of the influenza to some of the training resources, again, case studies, and you may recognize that there's a pattern behind all of those guidance documents, which we, again, came up with. So, what else we have? These are the finished guidance documents we have, and we're still working on for others as well. So, tuberculosis, TB sub-working group, working on a guidance documents for TB. We have foodborne pathogen sub-working group, working on the foodborne pathogens. We have phylogenetics one, and we have Oxford nanopore sub-working group. So these sub-working groups having separate meetings than the main one, and they are following the 5 steps that I explained previously. What else we do? We do knowledge sharing webinars. We had, especially this phylogenetic sub-working group lead this initiative. So we had introduction to phylogenetics. We had advanced phylogenetics with Zoe Dyson presenting. I think she's here. Also, Connor Meehan also gave us a presentation about tuberculosis. Kevin had a showcase of the best practices for pipelines, and we are aiming to continue with these webinars. So, you may think that we are bioinformatics pipelines and data visualization working group. Why not you're working on the pipelines, right? So, during the 2 years, we hadn't had a chance to work on the pipelines because it needs a lot of effort and a commitment from the community. But it doesn't mean that we are not going to do it. So we are, of course, after publishing our guidance documents as reference articles, we are trying to come up with maybe benchmarking data sets or benchmarking the common pipelines in the near future. And we are always open to suggestions, and I would like to encourage everybody who is interested to join to our working group as well. And I would like to finish my talk with empathy again. So empathy means to me sharing what you know and making it accessible in an equitable way. So always be mindful about accuracy, transparency, integrity of yourself, and technically create something usable. This is one of the things that we are trying to do in our working group, and make meaningful things for the community. And open source, of course, another thing that we are trying to achieve. And always there are constraints, challenges, but if we gather together, I think there's no challenge that we can overcome together. And thank you.
All right, thank you, Gültekin. Appreciate that. Always cool to see some of the work that we've done in the working group get highlighted, and also the various projects that keep that momentum moving forward. So our next speaker is Kathryn Murie, who will be showcasing her work on Plasmodium for Plasmodium genomic surveillance. So please welcome to the stage Kathryn for her presentation.
So my name is Kathryn Murie, and I work at the University of California, San Francisco. I'm also part of the Plasmodium Epi network, and it's that work that I'm going to be talking about most today. There's a QR code on there that takes you to the Plasmodium Epi website, and there you can see everyone who's part of it and some of the work that we do. So I'm talking about data standards and a curated kind of ecosystem around Plasmodium data. But it's not a new thing generating Plasmodium data. People have been doing it for a really long time, and there are currently 30,000 genomes that are public, and they have curated metadata alongside them. So it's not a new thing to be curating metadata around Plasmodium data. So I'm going to be talking a little bit about why now is a good time to revisit what a data standard for Plasmodium should look like. So why do we need a new solution for malaria? Why can we not just pinch something that someone else has already done? Malaria is a little bit more complicated in some ways. So malaria has kind of three components and three actors in the play. You've got the vector, the parasite, and the human that you have to consider. Plasmodium also has a really large genome when you compare it to things like viruses or bacteria, so you have to take that into consideration when you're doing some of your analysis. I'm not going to go too much into the genetics of malaria, but it also sexually recombines in the mosquito, and that diversity that you get from that is actually much higher than just from mutations. And then there's also polyclonal infections. So one person can be infected and have multiple different genetically distinct clones of parasites in their blood. So that makes some of the analysis we do a little bit more difficult to do. But we have lots of use cases for this data, and it can be really useful if we use it in the right way. So these are some of the use cases that are more kind of directed by NMCPs, National Malaria Control Programs, and then also some of them which are being developed in the research space. And this is one of the reasons that now is a key time to be looking back at our data standard, because some of these methods have evolved over the last decade or so and are now ready to be implemented, whereas they weren't before. So some of the key kind of phenotypes can be more complicated in malaria, so looking at like CNV calls and different drug resistance phenotypes. Sometimes you have to look at multiple SNPs, and then if you have a polyclonal infection, it all gets more complicated. So we've really been producing a lot of targeted amplicon sequencing data for malaria. This figure shows just 4 panels that are out there, but there are many more. And we've all said that we're targeting key regions of the genome, we're targeting drug resistance loci and things that people would be interested in monitoring, and we're also targeting high diversity regions. But somehow we all came out with different answers for what a panel should look like. And there's lots of data being produced. Targeted amplicon sequencing can be really sensitive, and it's also efficient and much easier to scale than whole genome sequencing. So that's why there's so much data being produced, and there's lots of panels being produced. And we can't merge that data together. Everyone's kind of gone off and created their own different panels and their own pipelines and standards around their panel, which means that for that data to be reused, it takes 80% of your time to try and merge that data together before you can even look at what it means. Another key component of this is over the last few years, microhaplotypes have become a really popular method. And I'm not going to get too into what a microhaplotype is, but if anyone wants to talk about them, I love talking about them, so come and find me. But microhaplotypes are basically targeting a really high diversity region of the genome and then keeping the information that you have about which SNPs go together and retaining that information, which you can do with targeted sequencing because you're sequencing from one strand of DNA. If you decompose that into single SNPs, you're losing a lot of information, and a lot of information that can be valuable for some of these interesting use cases like transmission intensity and connectivity between different infections. Unfortunately, the current solutions that are out there and that are standardized and everyone's accepting and plugging into their pipelines like VCF just aren't appropriate to be able to store this data. You either lose information or you cram it in some way and the file becomes massive and none of the downstream tools work with the VCF anymore. So there's lots of data being generated and a standard is really needed around microhaplotypes in particular and then also the metadata that goes along with them. And right now, not having that standard is meaning that lots of data is being produced in a really fragmented way and it's limiting us on how much we can reuse that data and create downstream analysis tools that people across different groups and projects can reuse. So it's kind of breaking FAIR principles right now. So this is kind of one of the proposed solutions that we have for a data standard around microhaplotypes. It's called the Portable Microhaplotype Object, and I'll refer to it as PMO going forward. This works for Plasmodium, and we were kind of motivated to create it for Plasmodium, but it's not specific to that, and we've shown that you can fit various different data types into a PMO. So we've put vector data into there, we've put E. coli data, and then obviously we've put Plasmodium in there too. We've been developing this for over 4 years, and it's a JSON structure, so it's relationally structured, and we're storing metadata and genomic data in one file format, so you're not in that situation where your bioinformaticians produce lots of genomic data and you have no idea what sample it links back up with. This also really helps with compression, so we've found that from taking lots of different data sets from lots of different groups, we achieve about 80-fold compression by putting it into this file format instead of storing it in scattered Excel spreadsheets in a folder. And we've tried to make this as accessible as possible by developing different tools around it as well. The tables that you see here, there is like data dictionaries behind each of these tables that describe each of the fields, and then those fields are also defined as required or optional. So the required fields are the minimum amount of information that you have to share with this data for it to be analyzable and reusable downstream. And then we haven't tried to reinvent the wheel. So over 60% of this data standard, the fields have been drawn from already existing standards, mostly the MixS standard, and we've really tried to just pull together the bits that we need. Okay, so some of the tools that we've developed around this, we developed PMO tools, which is a Python package. It's published on PyPy, so it's really easy to install. And then it's complemented with tutorials and documentation. And you can use that to build PMOs, to extract information from PMOs to filter, and then also really importantly to validate that your PMO is actually meeting the schema. We realized pretty quickly that not everyone handling this data knows how to code, so we wrapped this in an app that you can interactively drag and drop different tables into, and then we also incorporated a smart field matcher, so when you drop in your table, it will show you, here are the fields in PMO, and these are the ones that we think match up with your data, and then you can manually go in and change things if it hasn't done the best job. So that's PMO. And this is kind of how we see it fitting into the analysis workflow. So this is just kind of one part of a wider ecosystem, and definitely other data standards need to exist. The way that we've created this is super modular, so that hopefully when we come to downstream analyses, you can pick up the bits that you still need from PMO and get rid of the stuff that you no longer are interested in. We're not saying that you're going to share a PMO with a national malaria control program. It's just one step within the process. So after you've done kind of your different sequencing and then you've done your different bioinformatics pipelines, you would put all the information together in a PMO, and we found that this is where a lot of labs were sharing data between themselves. And then you can do all of these downstream things by having that PMO. So you can do a TES classification, you could look at different drug resistance phenotypes, and do all of that really interesting downstream stuff off of one core set of data. So the bright side is that a lot of the downstream tools have already been developed, and the downside is that they were developed before PMO existed. So they've all been kind of fragmented and developed off of people's own standards, which means that they're hard for people just to pick up and play and say, does this work for my data? And people just end up using what the bioinformatician next to them is using. So we've tried to start to harmonize around PMO, and we went and landscaped all of the different downstream tools that we knew of for Plasmodium, which was over 40 existing tools. We curated example data sets for them and created tutorials, and then benchmarked them against some standards for software development, like, can you even install this tool? So that all exists on the PHA4GE website, and then we've been undertaking some benchmarking, which will also go on the PHA4GE website, where we're using simulated data sets to say, okay, for this use case, this tool is currently performing the best, and hopefully when new methods are developed, we can add those in and see how they're performing. So we then went on to develop PGE Core, which is a set of wrapper scripts, which basically wraps around the tools that already exist and makes sure that they're all running off of the same data format and outputting the same data format, so you can really plug and play. And then we've used PGE Core as the building blocks for new workflows. A lot of the different utilities that we're using can be used in some of the different use cases. So for example, we have all of these PGE Core scripts, and then we might have a drug resistance workflow, which is an Xflow pipeline that threads together those different scripts, and a transmission intensity workflow that threads together those different scripts. But within that pipeline, you can choose which tool you want to use. So it's really interoperable, and it also means going forward, if new tools are developed, we can just pop those in and get rid of some old ones. So I just want to finish by saying thank you to everyone who's helped with this. This has been a massive community effort, and a lot of people who have volunteered their time to come to hackathons around this and worked really hard to kind of get this off the ground, and it's been really fun to do. Many of these challenges that I've been talking about, I'm sure, are not novel for malaria, and I'm definitely interested to hear from people, and I've been hearing great things throughout the talks from people in other fields, and we're not trying to reinvent the wheel, so if anyone has some solutions that they think could help, come and talk to me. My email address is on there if anyone wants to reach out. Thank you.
Thank you, Kathryn. It's always impressive to see 5 years' worth of work distilled down into a 10-minute talk. All right, and our next speaker is Noof Bani Khaled, who will be discussing her work on multi-drug-resistant Acinetobacter baumannii. So please welcome Noof to the stage.
Good afternoon, everybody, and thank you to be here this time. I'm Noof Bani Khaled from Universiti Putra, Malaysia. Today I will present a part of my genomic study for detection of rare ST547 among multi-drug resistant Acinetobacter baumannii in Malaysian hospital. So just through my contents in this presentation, I will go fastly about my research methods, results, and discuss those results in briefly. So, as you know, Acinetobacter baumannii is a gram-negative coccobacilli. It is multi-drug resistance, recognized as major nosocomial infection, and it is the major cause of hospital-acquired infections. So, according to World Health Organization in 2024, Acinetobacter baumannii, especially carbapenem resistance, as a first and most critical priority in hospital settings. If you look at the right, there is a map just show how carbapenem resistant prevalent overall the world. In Asia, Latin America, parts of Africa, and Middle East. If we go through just Malaysia, we can see there is increase, significant increase, especially for carbapenem, IMP, and MAM. So those carbapenem, around 60 to 70% increase since or from 2000 until 2016. This study, like a systematic review for many hospitals and medical centers in Malaysia. So the objectives of our study is to investigate the genomic antimicrobial resistance determinants, to find the plasmid profiles, phylogenetic relationship of Acinetobacter baumannii, those collected in 2021 in healthcare facility in Malaysia. I will go just quickly through my methods. Our methods to collect, first, we collect 55 samples. We took those medical records from the microbiology lab, and those data going just to take the Acinetobacter in general. So we didn't know what is the species specific for those samples. Then we did bacterial culture. So for those samples, we're doing two methods, genotypically and phenotypic. Then we compare the results. For the phenotypic, we use disk diffusion and MIC. We will not talk about it. And the genomic method. Through the genomic method, all samples go DNA extraction, PCR, 16 sRNA by Sanger sequencing, and go through the WGS. But not all those 55 go through WGS, just 37 samples. Those go through WGS. Why we choose it? We try to find the best DNA concentration and the phenotypic characteristics that we needed to further genomic. Those 37 go to nanopore platform. Then after we finish all nanopore, find 10 samples, low coverage. We try to repeat, but then we suggest to go through Illumina for short read sequencing. And after this sequencing, we go bioinformatic analysis to show to find the antimicrobial resistant genes. So for our results, the clinical and demographic data for those patients, we can see the highest percentage for male and the samples of the sources from tracheoaspirate, which means the respiratory system. And 85% samples from ICU, those who's in our isolates. For AST profile, we can see here most of the sample resistance to antibiotics, except I think one sample who is sensitive to amikacin. For the genotypic results, as I said, we identified all the samples, 55 Acinetobacter baumannii. And we just choose the 37 who's made our criteria to further WGS. And we can see here the coverage up to 342x, and the genome size around 4 million. Okay. Now what we found, for the ST, all samples by Pasteur, ST2. By Oxford scheme, just one sample, 547, and around 50% for the rest of samples from 1816 and the other from 1806. Okay. Now the antimicrobial resistance profile, same with mostly with the AST that we did. So you can see here most of the samples 100% resistance to carbapenem, resistance to gentamicin, to amikacin, and other antibiotics and aminoglycosides. This is just to show what is the resistant genes we found that distribute through the samples. So for aminoglycosides, most of them are resistant, as I said, but for beta-galactam, not all, just from each variant, one sample. This for all 37. For Plasmodium profile, it is interesting to see that 12 samples from those, 12 isolates, from those who go through nanopore, 12 are the same plasmid, and another 7 with other plasmid, I cannot see the name, and the other one, 117, also 5 samples. Those not found in literature. So we think that those plasmids, can we do further studies and further research in the future for those samples to see the resistance genes. The phylogenetic analysis, we can see here 5 clades for the Acinetobacter. The one we talk about, 547, just one sample, and others like clusters from 1806, 195, and also 1816 and 195. Okay. So to discuss further, this infection predominant among ICU patients and male patients, which is similar with other studies in Malaysia for other states, one in Kuala Lumpur and one in Terengganu. And most of the specimens from respiratory, which is also like other studies in China and near countries of Malaysia. Okay, and most of the resistant genes from aminoglycoside and beta-lactam. So WGS is still WGS driven by black oxa 23 consistent with its global dominance as other studies. And also the Odera, there is absence of RND efflux pump genes suggestion. There is alternative resistance mechanism. So all the samples under ST2, which is the same results for other studies in Malaysia. For the plasmid, I said before, there is no lectures that go through those plasmids, so we will do further study later to investigate it. For the ST547, you can see here, there is just 9 samples over the world. 7 that count, 5 in China and 2 in Malaysia. So we think that suggests there is maybe an epidemiological link between those samples. Maybe because some of Malaysian people from original Chinese, or maybe some traveling through countries. The phylogenetic tree, we can see there is similarities between those isolates. So we suggest maybe there is an outbreak during that period for the collecting isolates. So in conclusion, this study just provides and insight to the resistance, and also the clonal of dynamics of multi-drug resistance Acinetobacter baumannii. Still, WGS surveillance, can we investigate the resistant genes and find it? Also, for our detection of plasmids, we think also there is a horizontal gene transfer between those samples. Okay, so finally, just to acknowledge my committee in the university and the MLT in microbiology staff in hospital to assist us for this work. And hopefully we can do something new for the AMR. Thank you for listening.
All right, thank you, Noof, for that talk. I think it's always powerful to see when a lab can map phenotypic expression of the data on top of the genetic insights that you gleaned at that hospital. So our next speaker is Rachel Chomukama, who will be speaking about their work on synergistic drug combinations for E. coli. So please welcome Rachel to the stage for her presentation.
Good afternoon, everyone. My name is Rachel Claire Chomukama. I'm affiliated with the African Center of Excellence in Bioinformatics and Data Intensive Sciences. I'm going to present my work on titled Discovery of Synergistic Drug Combinations for E. coli from drug information, pathogen response, and disease microenvironment data. So as we all know that AMR is actually a global pandemic, in that it was predicted that by 2050 we shall have it is estimated that we shall have 1.9 million deaths to be attributable to AMR and 8.2 associated with AMR, which places it around, I think, second in the list of the top 10 killer diseases in the world. So according to WHO, the bacterial pathogen priority list, there is E. coli, whereby the carbapenem and the third generation cephalosporin E. coli was showcasing a high level of resistance, and which necessitates the development of innovative therapeutic strategies, since AMR actually inhibits since with AMR, most of the drugs that we have are actually not working. So this gives us room to actually develop new drugs. However, the traditional methods that we have for developing AMR, when we look at going in and developing new drugs, this actually takes a very long period of time, and it's also time-consuming and complex. But then, remember, AMR keeps growing, so we need to innovate new strategies that can match up the rate at which AMR is growing. This brings in our promising approach, which is combination therapies, whereby we repurpose existing drugs through combination therapy. That is looking at whether the drugs are, when used in combination, are they synergistic, additive, or antagonistic. This enhances treatment efficacy and reduces the likelihood of resistance due to the synergistic effect of the drug combination. But drug combination therapy has one big problem. We have very many drugs. So when you look at the possible combinations that we can have, there are millions of combinations. This is where machine learning comes in, and it helps us to identify what could be our potential synergistic drug combinations. So its purpose is to help us narrow down the combinatorial space. So our objective was to develop a machine learning model that can identify synergistic drug combinations against E. coli. So unlike the previous studies that have been done, they've been looking at these data sets in silos. That is the drug information, pathogen response, and disease microenvironment. That means that each model gives us the strength of a data set, and then we lose out on what the other data set would have offered to the model. So in this scenario, we actually explored the strength of all these data sets in order to give a model that would perform slightly better than what is already in existence. So we used 3 data sets. That is the drug information, which includes the specific substructures the drug is made of. Then this data set is in zeros and ones, showing the presence and absence of a substructure. Then pathogen response, which measures the sensitivity of a single gene knockout of gene strains in the drugs. Then the disease microenvironment, which shows us how antibiotic efficacy varies in the different metabolic conditions. So I'm going to take you through the methodology. So we had the drug information data that was in SMELS format. We processed it and got it in zeros and ones. That is the presence or absence of a substructure in a drug. Then we got the joint substructure profiles, since the drugs are actually used in combination. Then we also pre-processed the chemogenomic data together with the chemogenomic data of the metabolic conditions. So at the end of the day, we actually combined these data sets to form a complete an AI-ready data set, together with our target variable, which was showing us whether the drug combination is likely to be synergistic or antagonistic. Then we trained and tested machine learning. Then at the end of the day, we were able to predict synergy or antagonism. So these are some of the results that we found. These are the drug combinations that we gave to our model. What you see in blue are the synergistic drug combinations, and what you see in red are actually the drug combinations that the model predicted to be antagonistic. So remember, with machine learning, we can actually do feature importance. So when we actually do feature importance with the help of SHARP, these are some of the genes that the model considers to be antagonistic, or these are some of the genes that the model actually associates with either synergy or antagonism. So based on the genes that we used in our data set, we came up, through KEGG, we were able to do a pathway enrichment for the genes that we used. And those are some of the pathways that are the top pathways that we found to be relevant. So with feature importance analysis, we revealed that synergy is strongly associated with the integrated patterns across the drug properties, and equalize genes response metrics and microenvironment conditions, emphasizing the value of the multi-source drug fusion in drug discovery. So in conclusion, from what our machine learning models showed, the ensemble model was actually the best. However, at the end of the day, we have to use a model that can easily be explained or interpreted. So we didn't go with the best performing model. However, we considered the second best, which was the gradient boost classifier, because of its explainability capacities. Then the existing models, which is magenta and indigo, our model was able to outperform them. And for them, they had only put into consideration the genomic data. However, our model was outperformed by the cosine model, but for it only uses drug information data. So however much it performed better than our model has more features that are clinically relevant or biologically relevant compared to what it has. So the discussion is model performance. We can actually improve our model if we are able to get more data points, because one of the biggest challenges we found is that actually chemogenomic data is very hard to find. So we really encourage people, if they could share more data, would actually encourage development of better models and also coming up with better data-driven solutions for health. So, and also drug combination therapy is really, according to our model, it was really showing promising results, and then from this we shall proceed for animal and clinical studies validation. Thank you. And sorry, acknowledgement to my supervisors and the institutions. Yes.
Thank you, Rachel. Very important last slide. We'll let the PIs know that was included. All right, and our last speaker is Emma Hodcroft, who will be presenting her work on pathoplexis. So please, Emma, or sorry, rather, she threw me off because she went on the other side. Please welcome Emma to the stage.
So it is so exciting and also a great honor to be here today at PHA4GE to talk about pathoplexis. Oh dear, getting ahead of myself. A new database for viral genomic sharing that you've heard a little bit about already, which is in particular a community-driven solution for transparent and equitable viral genomic data sharing. Now, I'm pretty sure I don't need to tell this audience about the importance of data sharing. In fact, we've had a whole panel covering it this morning. But, of course, data sharing is really critical to doing science. And just to give a few examples, it is, of course, sharing viral genomic data that lets us do things like identify new variants or new mutations, to see how a virus might be spreading globally or within communities, and, of course, to develop things like vaccines, antivirals, treatments, and effective public health interventions. But despite the fact that we know that viral data sharing is critical, we also know that there are real challenges there and that data doesn't always get shared. Now, there are many reasons for this, and some of them I'm sure we'll discuss in more detail at this conference. But two big reasons are the technical difficulty and concerns about credit. For example, if you are someone who's more lab-based, or maybe you're a clinician, or you're just in the middle of an outbreak or an epidemic, if sharing data is too burdensome, it won't be at the top of your priority list. You won't do it. You might do it later, when things calm down or when you can get someone else to help you, but that doesn't mean that that data is available now when we need it to inform our response to an outbreak or to better understand a new pathogen. So we need to be honest about the fact that you shouldn't have to have a strong computational background just to do data sharing. It should be something that's easy enough to be accessible to everyone. And whether we like it or not, in academia and in research, publications are very much, sorry, my talk has a mind of its own. Publications are very much the currency of our livelihood. And so we need to be honest about the fact that if people feel like if they share their data, it's going to be scooped, that someone else will publish on it before they do, then again, they will not feel like they can share that data. And if that means that, of course, again, that data will be shared, it will get out there whenever the publication is ready, but we won't have it now, not when we might need it. And we may not like that incentive structure, but that's another talk. This is the incentives in the society, academic society we live in right now. Now, there are, of course, pre-existing viral sequence sharing options. I'm sure most of you have heard of them. One of them is INSDC that we already touched on today. What you may not know, which we did touch on a little bit, but maybe didn't explain so clearly, is that INSDC is actually a federation of more than just one database. So it comprises, at the moment, GenBank, DNA Databank of Japan, and ENA. And if you upload data, for example, to GenBank, it will appear on DDBJ and ENA as well, and vice versa. So all of that data is synced. There's also Curated that was founded in 2008 in response to the swine flu outbreak, where there was a big issue with people sharing data or not sharing data. They provide some protections to data sharers, and that has really encouraged more data sharing. So providing that sense of security is really important. Now, both of these existing options definitely have many pros. You've heard of them. They're well-established systems. They contain a lot of data, and that's really important because we don't want to have to go to 15 places to try and put together a data set. They have trust in the community. People are used to using them. They know how to use them. And of course, they've been around for a while, so they will probably be around tomorrow. We have trust that they will continue existing. On the other hand, though, there definitely are some cons. For example, there's really no flexibility in licensing. If you put your data on INSDC, in GenBank or ENA, it is completely open. And apart from the kind of ethical side, anyone can take that data and publish on it. They don't need to get in touch with you. Curated does provide some protections there. It cannot be used for everything. But if you ever want to change that, you need to re-upload it to INSDC. You can't change that license or have other options there. Getting data in and out can be another big barrier. If any of you have ever uploaded, for example, to GenBank, I've never met anyone yet that said that was just an amazing day that they really enjoyed. So that's a real barrier, right? If it's that difficult, it's not a big incentive to share. Getting data into GISAID is much easier, but for both of these, getting the data back out again can be a challenge, and that can be another hurdle that people run into further down the road. There's also limited interoperability, so you can't really move data between these two databases. You have to upload it twice, once to each of them. And then finally, even though both of these databases were really at the forefront of what they were trying to do when they were founded and have had updates since, to be perfectly honest, as a bioinformatician, they could be a little bit more computationally advanced here in this year of our Lord 2025. They don't have all the features that we would want to integrate into our pipelines and our analyses. And this is really the challenges that we are trying to solve with Pathoplexus. I don't have time to go into them all today during this short talk. I'm happy, of course, more than happy to talk about them. But in particular, we are aiming to be easy to use. That means both to upload the data to and to get the data back out to use in your analyses. We also have very clear data use terms that I'll cover more in a minute, but that outline how data can be used and how it shouldn't be used. In addition, we think that data versioning is critical. We need to be able to fix mistakes. We are not perfect. We make better versions of sequences. We get our dates mixed up. When we make those changes, we should be able to see clearly what it was in the past and how it's changed between different versions. And then finally, and perhaps most importantly, we are absolutely dedicated to transparent governance and being part of the community that we serve. As I already mentioned this morning, Pathoplexus currently supports 9 pathogens, and I'm really proud of that because that's up from the 4 that we launched with. So we currently support Crimean Congo hemorrhagic fever, Ebola Sudan, Ebola Zaire, HMPV, measles that we just added this August, Mpox, RSV A and B, and West Nile virus. And we're focusing on viruses that we feel, as I said this morning, don't have a home, those where we can make a difference by adding them to Pathoplexus, making that easier for the community. And we don't do this top-down, we do this in association with communities. So communities actually submit to us that they are interested in having their pathogen in Pathoplexus. Our executive board takes that into account and votes on whether to add a pathogen. Because we don't want to be running into other people's associations, their collaborations they're working with on different viruses and telling them they need to be on Pathoplexus. We want that to be something that's genuinely useful from them and powered by the users. I want to talk a little bit more about how data sharing and our data use terms works. As I've already mentioned, we've aimed to make this as easy as possible. So you can upload data to Pathoplexus by simply dragging and dropping a FASTA file and a metadata file into our website. That's it. You can even map the columns so you don't have to use our column names. If that even sounds a little bit too much, you can kind of see there's a second tab there that's a single sequence submission. You literally just fill in the fields that you feel like you're able to share and copy and paste the FASTA at the bottom. So even someone who's not comfortable with FASTA files or something can still share data. On the other end of the spectrum, we also have an API. So if you have a sequencing pipeline, you don't need humans clicking web pages. You can have your pipeline submit your sequences automatically. So we're really trying to cater to all of the user needs and abilities. Now, once you share that data, you have 2 options in how to share it, which I touched on briefly this morning. First, you can share that data openly, which is essentially the same as putting it on ENA or GenBank, and indeed, that's exactly what happens. So this means that anyone can use your data. They don't need to get in touch with you. What's different is that you can upload it to us this really easy way, dragging and dropping your files, and then we will put it onto INSDC for you. So we will take care of what for many people can be the hard part. We are a broker with ENA. What that means is that this will still be your sequence. You will be the authors. It will come from your institution. We can even add it to your biosample if that's what you like. So it's very much your sequence. We just do the wheel greasing part of getting it up into ENA. On the other hand, if you're worried about someone scooping your data, publishing on it before you can, but you still want to share it quickly for public health, you can choose to share your sequences as restricted use data. I want to really emphasize that's restricted use, not restricted access. All of the data on Pathoplexus is open and available. However, with restricted use, it cannot be used for everything. In particular, restricted use data cannot be used in publications and preprints without the explicit consent of the submitter. And when we say that, we mean that. It's not I emailed them 6 times and they didn't reply, so I did it anyway. They either need to be an author on the paper with you, or they need to send you a letter saying that they didn't want to be an author, but it's okay to use the sequences. Otherwise, you can't use the data. However, this is time limited, so you can have that data be restricted use for up to 1 year. It can be less if you want, and you can always release it early. That's absolutely fine if your paper comes out, for example. But otherwise, after 1 year, the data will become open. And this we see as a kind of balance. We think it's absolutely critical that people do have protection so that they feel like they have time to do an analysis and get a publication out while knowing no one else can publish. But we also know that long term, it is the most valuable to have data that can be used by others, because that is how we, for example, repeat and show that the same analysis can be done. Replication is at the heart of science. And also how we build on that data. If we have new variants next year, we need to be able to see how they compare from the variants from the year before. So we do think that having that data long term in a stable repository that's open is a good solution and balance here. Another thing I really want to emphasize is that Pathoplexis is designed to be part of this ecosystem, and it was fantastic to be able to talk about this a little bit this morning, because we're not trying to be a new island or a new castle, very much the opposite. We want to fit and be part of something that's bigger. So I've already talked about how data from Pathoplexis always ends up eventually on INSDC, so ENA and GenBank. We also do pull data into Pathoplexis from INSDC as well. This means that in Pathoplexis, you can access all of the same data that you'd get on INSDC, plus the restricted use data. So it's a very convenient way to do your analysis. Or even if you're just looking to use the open data, maybe you just find it more convenient to access that through Pathoplexis. I want to be really clear, though, we never hide that that's where we got the data from. It's always incredibly clear that it came from INSDC. You can always see the original accession number, and you can always link back to it on ENA or on GenBank. That's always possible. We do this just as a way of making it easier to get a hold of all the data you might be interested in. In addition, we wanted to make sure that, of course, we're empowering research. Now, all of the databases that I've talked about today support research and public health. That's why we have them as a database. But we also recognize at Pathoplexis that even though publications are still critical, they're not the only way we do science anymore. I'm sure many of you, like me, used other tools in the pandemic that were also critical. So you may have used analytic tools that were developed for SARS-CoV-2 that were enabled you to do very specific things. Or you may have used one of the many websites that developed to track cases, to track variants, to explain different mutations. These are critical for public health and for science communication, and we need to make sure that we're able to build on those tools because they empower not only public health interventions and informing the public, but of course further research as well. So at Pathoplexis, we have built a comprehensive API so that we can build these tools on top of that data very easily. So it can be done. People can make new analyses and new websites that build on top of that data. And one thing that makes us stand out a little bit is that at Pathoplexis, we do actively enable that the data can be redistributed. Now, of course, there's rules on this. You need to take the data use terms with you because it needs to stay attached to that restricted use data so that its protection stays. But at Pathoplexis, we don't see this as like, we can have all the data, it's ours, and you can't do anything about it. This is not our data, it's your data, it's the community's data. We are holding it and we are doing our best to be a good steward of that data and to enable good research. But if the day comes when we are not that good steward anymore, it's critically important that someone else could take that data, preserve the protection of the sequences, but put it somewhere else that the community perhaps prefers, so that data isn't trapped and so that no one is kind of held to ransom by Pathoplexis. In addition, as I mentioned, we also think it's really important to have versioning and community input into these sequences. So for example, if a submitter sees a mistake in their sequence, they can always update it, they can fix a date, or even have a new sequencing run that's maybe a little bit more accurate, and they can upload that and you'll see it as a version change. In addition, if members of the community spot a change, we can either contact the original submitter if it's a direct upload so that we can show them that maybe there was a mistake. We can even actually queue up the correction so that if they agree with us, all they have to do is hit agree and that correction will go live, which can be a lifesaver in an outbreak when you're kind of too busy to do a lot of corrections. Or if that data came from INSDC, we can actually make that correction ourselves, which can be a huge help because otherwise we all have to make that date correction or that country correction in every single individual analysis on everyone's computer. So let's fix it in one place so that we can benefit from that more accurate data. I also want to touch on as I said, our commitment to transparent governance and our commitment to being part of the community. We have an amazing board and membership. Pathoplexis is a separate legal association. We're not associated with the university. And that was really important to us because we want to be sure that it doesn't have to be attached to one person forever. Everyone in Pathoplexis could eventually rotate out. New people can come in, and Pathoplexis remains independent and a separate organization. We have a wonderful executive board, highlighted at the top, 5 people from 5 different countries, 5 different continents, my apologies, and I'm not just saying that because I'm on it, I swear. We also have an incredibly diverse set of members from over 10 countries and all sorts of expertise ranging from bioinformatics to virology to even legal ethics. And these members have real power. If the members don't like what we're doing, they can vote out the executive board and vote in a new one. So this holds us accountable, and it makes sure that we have an incentive to make sure that we are doing things that are empowering the community and that are what the community wants. And we recognize that may change over time, and that's good, and we should be able to respond to that, and we should be incentivized to respond to the community's changing needs. We also are committed to transparency, so all of our minutes and all of our resolutions are available online on our website. And we have, of course, monthly meetings with the PHA4GE community, so we are part of Working Group 6. We meet once a month. Please join us if you're interested. And this allows us to have an incredibly valuable way of finding out what do people want, what are suggestions, maybe a problem or a feature that's missing. So this is an excellent way for us to stay in touch with those who are using Pathoplexis. I want to just highlight, even though we've really only been running a year, we are so honored to have been able to play a role in outbreak response already. So for example, both for the earlier outbreak, Ebola outbreak in Uganda at the very beginning of this year, and as Placide talked about this morning, the more recent Ebola outbreak in DRC, both of these outbreaks chose Pathoplexis as the place to share their data. So not only did they do an incredible job of sharing that quickly, they trusted us with sharing that data as restricted use as soon as they had those sequences. In addition, we are the database that had received the most mpox sequences in 2025, and many of these also come to us as restricted use. And to me, this really highlights that there is a need for different ways of sharing data in the community. People are seeing that this fills a gap, and in particular, they are seeing that having these time-limited protections enables them to be able to share faster while still knowing that a publication can follow. And I'm really honored that these people have chosen to share this data with us, because I think it does show that we can encourage data sharing by creating an environment where people feel safe to share. One of the last things I want to touch on is that we're also very aware that, of course, data sharing during kind of peace times is very different from the data sharing that we might need if we have another, or if and when we have another pandemic. And Pathoplexis, we're really flexible. We have designed this so that we can respond rapidly. So within minutes, we could add a new virus and have it live to receive submissions in, for example, a new pandemic scenario where that was critical. It might be fairly basic at the start because we may not know much about a new virus. That's kind of how novel viruses are. But what's also good is that as we learn more, as we get annotations, as we get reference genomes, clades, these kinds of things for the virus, we can add those and they will retrospectively be applied for the samples. But what's critical is you can share that sample as soon as you have it. We can start that precedent early with a new virus, and indeed making that restricted use if that's what people feel like they need to share that sequence. I don't have enough time today to talk about the code behind Pathoplexis, but I want to mention it briefly because it's also really important. So we've also built the code that runs the database of Pathoplexis. It's called Loculus. It is, of course, open source and transparent, like everything that we do. It's also highly flexible and pathogen agnostic. And so we're really hopeful to develop Loculus into another kind of data sharing solution, where this could be, for example, something you could use to store sequences at your lab, at your university, or indeed even at your country level. And what we'd like to see is, of course, have that be easy ways to then share from your local Loculus up to Pathoplexis, or even to different Loculus instances, perhaps your collaborators. This is something that we're still working on, having these local releases be something that we're really able to say to the community we're ready to use, but it's something that we'll be focusing on in 2026, so please stay tuned for more news on that. So finally, I really hope that today I've been able to give you this brief introduction into something that we are so excited that we've been able to bring to the community. A data sharing solution that helps to empower sharing, to make sharing faster, easier, and hopefully also more equitable, And of course, this then enables all of those downstream things, enabling prospective pathogen research to keep us safer from what may come tomorrow, and also enabling better surveillance so that we have a better idea of what's out there and how to reduce that health burden. My talk is really getting ahead of me. Finally, if you have any questions, there are many Pathoplexus members here. There are many Working Group 6 members here. Please come and find me. I will be happy to chat. You can also talk to my Working Group 6 co-chair, Arthur, or the Pathoplexus co-founder, Karan, who's also here. And we have wonderful stickers. So if that's what you need to come and speak to us, please come and ask for a sticker, and we're more than happy to talk to you and answer your questions. Thank you so much.
All right, thank you, Emma, on that talk. Really appreciate the democratic lean you guys are taking with Pathoplexus. Feels very much like a resource for the community, by the community. And now we have brought all panelists back up to the stage. So we've got about 20 minutes for a Q&A session. And if you have a question, please raise your hand and a mic will be brought to you there. I'm supposed to join you guys. Test. Mike, can you guys hear me? All right, solid. All right, so as the mics are getting passed around, I can kind of open the session up and maybe start with Gültekin on your talk on the Pipes and Viz working group. You mentioned something that Jamie and I also saw in the working group, in that it's called Pipes and Visualizations, but we didn't really see a need to be developing pipelines because there's really so many different pipelines out there. The major challenge in the community is having resources to guide selection and implementation of different pipelines. But what you mentioned that you guys are walking into is a huge need in the space of maintaining pipelines. So what sort of role do you see the working group playing in terms of maintenance of pipelines and workflows for pathogen genomics?
First of all, thank you, Kevin, for the question. I would like to thank you again, while you are next to me, about the legacy that you handed over to us. It was really nice to be a co-chair of the Bioinformatics Pipelines and Visualization Working Group so far. So the initial idea was taking the legacy of you provided to us and try to take it a little bit more forward. So I know we had some validation data sets and the pipelines that we created in the previous era. But in the near future, we will mainly focus on phage validated pipelines. These are all thoughts that we have, some of the projects that we are in mind. So what I mean by that, like there are many pipelines out there, and you can go and work with it and have some results. But reliability of the results vary between pipelines to pipelines, and there is no standards. I mean, we have 10 simple rules for bioinformatics pipelines, but there's no simple pipeline, there's no simple results that you can get, and sometimes you may question the results that you're having. So in phage, we may come up with a solution to that, which can be phage validated pipelines. We can use the 10 best practices that we have, but we can create some benchmark data sets, and with the, of course, help of the community, we can validate these pipelines and make everybody sure that this pipeline is working very well, and of course it will be backed by some articles as well, of course. But yeah, I think in the near future, some of the projects that we are planning to do.
Very cool. Nice, great to hear. Just checking if there's any questions across the audience. I think we got some here, please.
Thank you. Hi, Khaled Youssef from NICD South Africa. My question to Emma with regards to the new platform, looking great. I have a question about the public health use, and would it be possible to consider trying to add a component of translation, the data that we generate and submit, and how can it be translated to public health eyes? And my understanding that most of the time we focus from academic perspective or scientific discoveries about new knowledges. But when epidemiologists look at the data, whether it's lineage 1 or lineage 2, with the presence of these mutations, for them it's not complete. What is complete is to understand what are those lineages meaning from clinical surveillance perspective. And to my knowledge now, there is no database or no platform can put the guidelines for that. We're actually doing it one by one in our meetings within our institutes. I come from Public Health Institute, and this is a main challenge that we have, because every time we have to sit around the table, discuss specifically during the SARS-CoV-2 pandemic, and also from, because I also work in wastewater space, and this is also another challenge for the wastewater findings. That's how do you translate it? How do you go beyond the idea of just doing the work and publishing the article? Because at the end of the day, you're looking for the impact.
So that's a great question, but I'm afraid you're not going to love my answer, because part of what's very interesting with the work that we do at Pathoplexus is, as much as deciding what we are going to do, it's often also deciding what we aren't going to do. And one of the things that we, at the moment, at least the executive board at the moment, has decided that we don't do is provide direct analysis or interpretation of the data. And this is really because we don't believe that we should be the ones that by default say what the analysis is or what the interpretation is. Science is done by many people, and who are we to say what the analysis is? Now, the other flip side of that is that even though we don't think that Pathoplexus is the one to say, this is the analysis, trust us, we know that that analysis, and as you say, even other resources, is incredibly valuable. So what we have with the API is a way to allow people to link from the data to tools. At the moment, we just have 2, but we're very young, and this isn't something that has to be developed by Pathoplexus. We can have any tools that maybe even people in this room, if you would like to see an mpox report format or a website that shows particular variants in another one of the pathogens we support, you can build that and we will link to it as long as you can accept that data coming in a very straightforward format. So for example, what this would mean is that you or maybe someone else could build a kind of mpox, for example, report format that explains in more friendly language what the lineages are circulating, what that means, what mutations they have, what the interpretation is, and you could link that out from Pathoplexus. What's nice about us not doing it is that also means if someone else wants a different report format or they disagree with what you're saying about the lineages, they can also make something that does that. So it means that we have a more democratic way of building the tools that we need, but then making sure they're available for everyone. So I hope that's a bit of a balanced answer, that the answer is, well, we're not going to do it, but we want to enable it.
Emma, as a follow-up to that question, though, you know, with other databases, like, for example, GISAID, when you submit SARS-CoV-2 data, they do some kind of QC or original check. When they're looking at the data, so that kind of allows them to infer lineage, for example. And then that's actually the lineage that's included in GISAID. Is that not the Pathoplexus approach? Yeah.
Yeah, so sorry, I should clarify. We don't do the more detailed analysis, but we do basic QC. So we do some quality control checks. We also do things like we list all the mutations between your sequence and the reference sequence. That includes amino acid translation, so amino acid mutations as well. And then also things like the lineage assignments and this kind of thing. And we're going to add more of these QC checks in the future to make it easier for people to, for example, exclude sequences below some certain threshold. So I guess that is analysis, you're right, but it's really just kind of things that help you to understand that sequence, not taking all the sequences together and doing something more in depth with it.
Fair. Please.
Yeah, thank you very much. I'm Samuel from MRC Gambia, but I'm speaking from experience because I work in Nigeria. My question, or maybe appeal, is to Emma. In your presentation, good one, but you mentioned the restriction period to be 1 year. And then I'm thinking, as someone that is coming from a low resource setting where priority is not given to publication. Now, I have the data, but I know that within that 1 year, I might actually get it published. So I will still have to keep it. So the question is this cast in gold, or you can just extend to like 2 years so that people that have, not everybody have the money to generate the data. You source for the money yourself from salary, student, paying for that data, and then you get the data. You still have to look for the money to do the publication. And then you are saying 1 year. Within that period, the salary is not enough to do that publication. So maybe you might have to consider that in settings too. Thank you.
this is something that we thought about a lot when we were starting Pathoplexus, and actually it's one of the things that we drew on the phage community to ask. How long should the kind of embargo period be? Should it be 3 months, 6 months, a year, 2 years, 5 years? And we talked about this a lot because we recognize that, to be fair, even if you submitted the publication the day after you got the sequences, with the review process and burdens that we have in journals today, you still might wait a year before you get that paper out. What we came up with this 1 year was essentially the consensus from everyone that we spoke to. At the moment, that is fairly set, as in that's in our rules, but it's possible that the EB and the members could decide to change this. But I guess I also want to put a little bit of emphasis on the other end, that I completely emphasize that it's a disadvantage for many countries that they cannot do analyses as quickly as some other countries, and that disadvantages people for publications. However, I think that the longer term answer is we need to really scale up how much we are funding this and how much we are expanding education and access. Because one of the biggest things, in my opinion, to enable data sharing is enabling data analysis. Because if you know that you're going to be able to get a paper, or at least a preprint, if you're worried about the review system, at least a preprint out quickly, if you know you can do that, you feel empowered to share. So it's not just about saying data sharing, data sharing, data sharing. It's about backing that up by making sure we have the education, the workshops, the training materials so people know how to do the analysis, and then funding, not just generating the data, but making use of it in the countries where it's being generated. And my hope, I know I'm being a little optimistic here, is that we start doing this better so that everyone can publish in a year.
Well said. Kathryn, can I ask you a question? You had the talk on the different Plasmodium approaches. I think one slide that was kind of surprising is that there's a targeted amplicon approach, but there's competing panels. I think you had like 5 or 6 different panels. Can you explain what's going on there? Is it that so many people are interested in different genes, or are these largely overlapping with maybe just minor differences in primers?
I think this comes largely from the funding structure and what motivates projects, and a lot of the time the funding is, here's funding for one specific project, and then you build a panel, you build your pipeline, and then you analyze your data and put that data out. And the funding structure doesn't really motivate people to use the same panel. Although there are lots of groups where panels are being developed and there's multiple people using them, and that is becoming more common. It has just been the way that the panels have been expanding as people have jumped on kind of the bandwagon of making them.
Looking through, please.
Hi, the question is to Rachel, and I was actually fascinated by your one of your drugs, Novobiocin, and we actually picked it up when doing some in vitro work with TB. And what we were struggling to find is examples of how it is used in either human cell lines or perhaps even something more complex. And where we got stuck was that its primary uses in veterinary science. I was just curious if you had any kind of idea as to how its utility or its safety would work in a human cell line, for example.
Do you mind repeating that? Yeah. Do you mind repeating the last bit there?
Yeah. What I was curious with Novobiocin, and you had it as one of your top candidates. You had Novobiocin as one of your top candidates as part of the candidate screening, and you were trying to figure out how can you actually deal with antimicrobial activities. So the machine learning identified these metabolic biosynthetic enzymes.
Okay, I think you're talking about the pathways.
Correct, yes. The biosynthetic pathways.
So for now, we are still on the next stage of exploring what the different pathways do and how they actually contribute to the synergy of the drugs. Yes, for now, we have only done the machine learning, getting the drugs, and then also identifying the pathways from the genes that we actually used for the training and testing of the model.
Rachel, as a follow-up to that, with those combo therapies, is there an established benchmark literature of combo therapies that you could, validate what your machine learning models have proposed, or is this something that you got to go in the field and test?
So the machine learning model was, we benchmarked based on the pre-existing, some of the combination therapies that were either known to be synergistic or antagonistic. That is the test set. Our test set was basically drugs that are actually known to be synergistic and antagonistic, also the train set. But then for the graphs that I showed later, those are drugs that are, for now, we could call them novel combination therapies.
Got it. Thank you. We got time for one more question. Yes, please.
I would like to ask a question from Nooshi. No?
Noof.
Noof.
Noof, yes.
Okay, sorry. You were showing on one slide about the genotypic results for the antibiotics, and most of them, they were showing 100%. Okay, so did you check on the phenotypic side? Because we didn't see that slide where you were showing the phenotypic and the genotypic. So under the genotypic, yes, we can see it's 100%. But if you see on the phenotypic, maybe it wasn't even 100%. So did you do the phenotypic and the genotypic on the same samples that you were doing? Thank you.
Thank you. But in the beginning, I show you the AST phenotypic for those samples. All are resistant except one samples. Those for 55 samples. When we go through genotypic, we just did for 37 samples. And those the same, resistant. But we did by, I talk first that I did phenotypic by 2 AST. One by MIC for cholesterol, and the other one for disk diffusion, for the routine antibiotics resistance. So all the samples resistance to those the same in genotypic. But we can see the genotypic why 100%, then 49%, and so on. Those for just the 37 samples. But those 55 all done by phenotypic. And just for cholesterol, because I just talked lastly, that this cholesterol we already published in another conference. We talk especially for cholesterol, because when you find some samples that resistance to cholesterol is not easy. So most of them should be susceptible. But this sample, yes, resistance to cholesterol just phenotypically. So we have to do phenotypic, then to check by genotypic.
All right, thank you all. Please give a round of applause to our panelists.